Okay, right. I think while we're just letting a few people in, I'd like to say hello and welcome to this Imaging One World talk, which is with Lucy Collinson from the Crick Institute. And Lucy's been running electron microscopy facilities since about 2004 and has actually run quite many successful projects, and one of the most enjoyable people I know that's an EM person to talk to, and has some interesting sidelines in outreach and her Zoomverse. So anyway, over to Lucy, a brief introduction for you.
Thank you very much, Alex, and thank you very much for inviting me to give one of these One World talks. It's really exciting to be able to share our current work with you, and I'm going to use today as well to introduce some of the lab to you and to show you what they've been working on as well. So just a quick note that if you have a camera on your phone, then if you can have it ready, then there's going to be some QR codes coming up with links to take you straight through to websites for various things. So to start with, what is correlative light and electron microscopy? I'm aware that we have an audience with a mix of different expertise here. So correlative light and EM is a way of tying together information from fluorescence microscopy images. So we might have a fluorescently labeled protein, and then in light microscopy, we might be able to see the architecture of cells here, neurons growing on cover slips. But it's not until we track the same region and image with electron microscopy that we can see the really fine ultrastructure. So here, a single synaptic vesicle communicating across a synapse. So it allows us to do a couple of different things. One is to target particular structures in cells and tissues, and the other is to localize proteins in a similar way that you might do with antibodies and immunolabeling. This is a beautiful sketch from Chris in the lab that just highlights that there are many different ways to do CLEM. Sometimes people ask me which CLEM microscope I recommend, and there isn't a single CLEM microscope that will answer all questions. And instead, CLEM is really a pipeline. And the first type of CLEM I'm going to talk about today is volume, and then later I'm going to talk a bit about precision, but I'm not going to talk about cryo today. So one of the pieces of work that really inspired me early on when I was starting to understand the potential of volume electron microscopy was the work of White, Southgate, Thompson, and Brenner. So this was published in the 80s, and the subtitle of the paper was The Mind of the Worm. So they took individual C. elegans, they prepared them and embedded them in resin, and then they sectioned them in a process you can see behind me. So that's embedding the sample in resin and cutting it into ultra thin sections into about 10,000 sections, and imaged each section in order, and then took all of those images and stacked them up into a volume so that they could reconstruct the wiring diagram of the nervous system of the worm. And things have progressed hugely in the last 40 years. I can't believe the 80s was 40 years ago, but that's another story. So back then they were using serial section transmission EM, cutting sections and picking them up onto grids and then imaging them in a transmission EM. And then followed serial electron tomography. So similar sections on grids, but you can increase the resolution within each section. And in the last 10, 15 years, there's really been this explosion of the serial block face techniques. So these are now based on the scanning electron microscope. And instead of slicing sections and picking them up onto grids, the cutting is done in situ inside the scanning electron microscope. In the serial block face, that's done with a miniaturized ultramicrotome and a diamond knife. And in the focused ion beam, that's done with a beam of gallium ions, which sputter away very thin layers of material from the sample surface. So cutting up here is around 50 nanometers thick, and down here is about 5 nanometers thick. And then we have array tomography. So now we're back to cutting with a diamond knife and picking up the sections, but this time onto much larger substrates, usually silicon wafers or ITO coated glass. And again, these big sample carriers can go into a scanning electron microscope, and then the sections are imaged sequentially. And then you have improvements in cutting and start to automation of sectioning so that we can pick up thousands of sections at a time. And for this, we now have grid tape TEM and Tempca TEM, which are imaging thousands of sections through the transmission EM. And then improvements in the ion beam and the block phase techniques. So coming from focused ion beam, we now have plasma fibs and gas cluster ion fibs, which can mill much larger faces, but still maintain that very thin cutting. And then we have tools like laser milling, which can help us to prepare samples for the ion beam techniques. And then we have the multi-beam array tomography. So this is where the wafers are put into a scanning electron microscope and imaged with the electron beam, which is split into multiple parallel electron beams, which means that you can image a large field of view, but maintain very high lateral resolution. And then we have various associated techniques. And one of the main ones is correlative imaging. So when we're talking about volume EM, you're normally starting with an entire cell, which is still large for electron microscopy, or even a model organism or a piece of tissue. Then the field of view in EM is still usually much less than a millimeter. And so that means that within your entire sample, you need to target the region of interest and trim down so that you can image that small volume. So it's like finding the needle in the haystack in EM. And micro-CT and synchrotron X-rays are a massively exciting technique for doing that, because they allow you to see inside your sample non-destructively before you cut into it. And so we've gone from the mind of the worm in the 80s, which was a 10-year effort, to the current state of the art, where the connectome of organisms like Drosophila and zebrafish are being mapped by institutes and consortia like the Fly EM effort from HHMI and Google. And the next big challenge, which was presented in this really nice paper in Cell, is looking at the mind of the mouse. And just to give you an idea of the challenge there, they put forward this beautiful analogy. So if the mind of C. elegans is the width of an airline seat, then mapping the mind of the fly is in scale similar to 6.5 Boeing 747 airliners, but moving to the mouse then takes you to the entire distance from Boston to Lisbon. So there are huge challenges still in reaching large volumes, but at very high resolution. And one thing that we really can't forget as microscopists is if you put a rubbish sample into the microscope, you'll get rubbish images out. And sample preparation is key. And in electron microscopy, sample preparation is pretty challenging. So just for those who aren't familiar with EM, for volume EM techniques, we're normally handling samples embedded in resin. We're normally using a chemical fixation, using paraformaldehyde and glutaraldehyde to cross-link the proteins and stabilize the sample. And then adding heavy metals like osmium tetroxide, and particularly for block phase techniques, we're also adding uranium and lead salts to put plenty of metal into the sample and make it conductive. That also cross-links the lipids in the samples, so it further stabilizes the biological sample. We then dehydrate, so we remove the water and mix with our resin before baking it or polymerizing it with UV. So normally for volume EM techniques, we're going into an epoxy resin because it behaves better in the scanning electron beam. And those resins protect the sample from the vacuum, but they also give us good sectioning properties so that we can cut ultra thin sections down to, say, 50 nanometers. So just looking in a little more detail at one of these techniques, this is our serial block face SEM at the CRUK. We have a Catan 3 view on a Zeiss scanning electron microscope. And the technique works with the diamond knife passing over the sample surface and cutting a section and then discarding it. And then the electron beam images the cells or the tissue embedded in resin. And then the diamond knife cuts, and then you image, and that means that you can automatically collect a stack of images, usually around 1,000 in 24 hours. So here you can see a scroll through of the images collected from a pellet of HeLa cells embedded in resin. And one thing to note is that almost all of the volume EM techniques, and indeed the X-ray techniques, result in a stack of images that scroll through the volume of the sample. So actually, even if the data collection is different, then you end up with these blocks of data that you can slice through virtually. And we don't just use these techniques for cells, we use them for tissues. So on the left, we have an example of a piece of liver tissue. And on the right, some mouse retina looking at the lumen of blood vessels during the process of blood vessel fusion. If you are interested in volume electron microscopy, and this is the first time that you're hearing about it, then please grab your phones. So in the last few years, the volume EM community has come together. It's a very friendly community and very motivated, and has started both proposing infrastructure to support bioscience, but also importantly, networking nationally and internationally to create tools and to bring the community together and to advance the technique. You can find a lot of information on our new Volume EM community website. And if you go to the working groups page there, you can meet the co-chairs of various groups who are looking at community outreach, sample prep, data, training, and infrastructure. And if you're interested in getting involved in any of those, then you can submit inquiry through the website, or you could join us this Wednesday when we're having our 2021 Volume EM Town Hall meeting. So there's still time to register. Again, this is a different barcode. It will take you straight to the events and registration page on the website. And we have a full day meeting looking at all of the activities that have happened in the last year, and also some talks from international experts on the cutting edge in volume EM. So we hope to see you there. So the question now is, we can collect beautiful three-dimensional high-resolution ultrastructural data using volume EM, but how do we make that a correlative experiment, and how do we find that needle in the haystack, the small region of interest in the sample? So I'm going to give you an example of some recent work which was done by Marie-Charlotte in the lab with Nagisa Yoshida and Serge Mostolvi and Eva Frickel, who were looking at zebrafish and whether it can be used as a model organism for the in vivo study of parasites. So here you can see the zebrafish hindbrain. You can see Toxoplasma gondii, which is a parasite infecting the hindbrain. And you can also see macrophages, which are labeled in red. So just as a general principle for correlative experiments, we normally start with fluorescence microscopy and would use whichever type of fluorescence the research group has optimized for their sample, whether that is wide field, confocal, maybe light sheet or super resolution light microscopy. So in the zebrafish work here, the hindbrain has been cut into 100 micron thick sections using a vibratome and then imaged in, this is either confocal or airy scan. So now you've already cut down your volume from the entire model organism, and you have some very nice resolution on the individual parasites and the macrophages. So MC would then take that information and use it to target. So I'm missing out some detailed steps in the protocol. I'll come back to that later. Use that to target where the individual parasites are, and collect here serial block face SEM data from those regions. So here in each of these squares in the middle, you can see the parasites. And there's a couple of things to note here. So first of all, because we have volume data, you can scroll through and you can see that in the first one, there is only one parasite. In the second, there are two, but they're joined together because you can scroll through and you can find the connection. And in the third, there are 2 parasites, but there's no connection. So they've completed a division cycle. And actually, you can start to be quantitative because here MC's targeted multiple parasites and collected 3D data through each. And so you can see multiple single parasites. You can find multiple parasites that are still connected and undergoing division, and then some which have completely divided. Now, the referees came back, so that was submitted as a paper, and the referees came back and said, well, we can't see the individual membranes in this data. So serial block face can reveal organelles, but you can run up against resolution issues compared to transmission EM. And so they requested higher resolution to show the parasitophorus vacuole, which is the host membrane that is formed to contain the parasites inside the cells. But we still needed 3D data. And so what MC did was use serial block face SEM as a smart targeting tool. So here again, we have the vibrotome sliced with the fluorescence, and this is our region of interest with a macrophage containing 2 parasites. So she embedded the sample, put it into the serial block face SEM, and then whilst watching the images being acquired, she tracked down until she found the region of interest just above it, and then transferred the sample over to the focused ion beam scanning electron microscope. And here you can cut, rather than 50 nanometer sections, you can cut 5 nanometers. So here's the macrophage and the 2 toxoplasma parasites coming into view. You can see nice membrane whorls, which are characteristic of the macrophages. And we can also look at the data in any direction because we have 5 nanometer isotropic voxels. So we can match up the FibSem data to the original fluorescence data. And MC found a couple of things. First, that you can see mitochondria sitting very close around the PV membrane, which is similar to what happens in mice, which suggests that the model is good in zebrafish. And you can also see, marked by the blue arrows, the PV membrane going around the outside, and also that there are breaks in that PV membrane, which suggests that the parasite is able to escape. And that FibSem data was collected by Chris in the lab. So that work was done in zebrafish, so that's an entire model organism. I'm just going to give you some recent examples from the lab, because every correlative experiment is different, and every correlative experiment is designed to answer a specific biological question with a particular sample, which you need to image with a particular field of view or volume and a particular resolution. So MC imaged a very similar sample from Eva Frickel's lab, but this time looking at Toxoplasma gondii in THP1 cells. So those are monocytes growing in culture. And in this case, using serial section transmission EM, because the sample's a lot smaller, and also to get much higher resolution of the PV membrane. MC also did something similar, working with Catano Rise Souza. So here we're looking at cells that are taking up DNGR1 labeled membranes and breaking them down. And you can see that this vacuole containing the DNGR1 membranes actually has a break in it. So again, we're looking at high resolution to find breaks in organelles. Whereas with Azumi Yoshimura's work, so Azumi has just moved on to run her own facility in Japan, which is very exciting. She was working with Alex Gould in the field of metabolism, imaging Drosophila nephrocytes, which are much bigger cells, and looking for endocytic structures. And this work is done with serial block face SEM, as is a second study where Azumi was looking for neutrophils escaping between endothelial cells in a blood vessel in response to inflammation with Susan Norshaw's lab. And this is also bringing in autophagy labels for ADTG5. And then moving to a Fib project, but at a much larger scale, Chris recently had this collaboration with Vasilis Paknis, published looking at zebrafish intestine and looking at cells which were putative glial cells and looking for their structure in EM. So all of this is to say that everything has to be custom designed according to the sample, and that in every case here, the fluorescence labeling was not enough to be able to answer the biological question, and the underlying ultrastructure and or the context was required to understand the biology. And the other thing is that all of these data that you see in papers, so you don't normally see volume EM in the title of a paper nowadays, because actually it's established in lots of the large EM labs to some extent, whether it's serial section TEM or serial block face SEM, or more rarely focused ion beam SEM. So you have to dig into the papers, and then you also have to look for the movies, because this data is massive and it's 3-dimensional and you can scroll through it. And I hope you can see these videos okay. And actually here we have some really nice moving arrows in the data to show the cells of interest. So what was on my workflow, but I didn't really mention so far, is that we frequently drop an X-ray imaging step in the middle. So if we go back to the zebrafish, you can see how amazing these X-ray images are. So the sample turns 360 degrees in the path of the X-rays, and you take multiple images and then stitch them, reconstruct them into a volume. So here we can see the zebrafish eyes, the zebrafish hindbrain, and in 2D slices, you can see the somites, the retina, and you can even start to see the individual toxoplasma parasites. But when we use X-rays, it's not always in a workflow with light, X-ray, and electron microscopy. X-rays are actually incredibly useful in their own right for imaging soft biological tissues. And Anne Weston in the lab has done a lot of work on this, looks after our Versa 510 micro CT system. And she has both done testing for sample preparation to find the optimal way of preparing and supporting these samples, and has been looking at how far you can push the resolution. And I still find this really incredible. So she put a small piece of resin containing individual HeLa cells into the system, and imaging at high resolution, you can see the individual cells, you can see the nuclei, you can see chromatin patterns, and you can even start to see mitochondria. So this system is imaging with about 1 micron resolution, and it just blows my mind that you can see this much detail, and that it's also non-destructive, so that sample is still intact and you can go back and cut into it for volume electron microscopy later. And that's just what MC and Alana have been doing, and actually many people in the lab are incorporating micro CT steps into their work. So MC finding putative ependymoma tumors in mouse brain to then target them to look at hallmarks of cancer. And Alana finding these fiddly little carotid bodies so that she can cut sections and look for secretory granules, investigating the regulation of hypoxia. If you're interested in learning more about hard X-ray imaging of biological soft tissues, this talk also came at a good time, because on Thursday this week, you can connect in to the second virtual meeting on hard X-ray imaging of biological soft tissues, which was organised by Carlos Bosch at the CRUK with Andreas Schaefer and Liz Duke, who's now a team leader at EMBL Hamburg. And you'll hear from a range of experts who are using X-rays in different ways to get at the structure of biological samples. So please do go ahead. If you can't get onto the Eventbrite site today, then try back tomorrow. I think it was closed, but it's going to open again in case you want to register. So the elephant in the room, of course, is the amount of data that we're producing. And that gives us a huge number of problems in handling the data, in moving it around, in sharing it with people, in visualizing, in analyzing, and in quantifying. And one of the main areas that is a bottleneck right up front is segmentation. So extracting three-dimensional structures from the stack of 2D images so that we can visualize and analyze them. So this is Anne in the lab, and she's using a touchscreen to draw around the nuclear envelope in a single image. To do that in a stack of around 200 to 400 images going through a single cell would take an expert a week. And we just chuck out data at a rate which means that we can't do this, but it is critical for interpreting volume EM data in fields like blood vessel fusion, differentiation of stem cells into neurons, and in the maintenance of immune responses. So if we hadn't done the reconstructions in this case, which took 4 months, about a month and a few weeks, then we wouldn't have been able to answer the biological question. Oops. So given that we're so limited in experts to do the segmentation, Martin Jones, who runs our microscopy prototyping side of the lab, had the idea to go to citizen science, so to use a much larger number of non-experts. And with Chris Lintott, who runs the Zooniverse Citizen Science platform, and Helen Spears, who is the Zooniverse biomedical lead and now a CZI imaging scientist between Oxford and the Crick, set up this project EtchAcell. So again, if you've got your phone out for the QR code, this will take you straight through to the Etchaverse. So EtchAcell is now expanded to the Etchaverse, and we have multiple projects online. The idea behind all of them is similar. So we feed out these stacks of images to the general public, and we ask them to draw around features of interest, like the nuclear envelope. So it's, as well as being useful for us in gathering ground truth data, it's also a great public engagement tool and is being used for education in schools. And so the first project we had was on the nuclear envelope. So that was EtchAcell going nuclear. We had more than 6,000 people drawing lines and more than 130,000 classifications. And this is just showing you all of the lines drawn on a single image, which you can see are of varying quality. So we feed out each image to between 15 and 30 citizen scientists so that we can get some consensus. And the result is that the aggregated citizen science data looks very similar to the expert. And from this aggregated citizen science data, we can train a UNet to automatically segment the nuclear envelope. Turns out that this aggregation step is the really difficult part, which actually took more than a year, probably a couple of years to get a grip on. So to automatically find the right line from all of these varied contributions. The results of the model are really impressive. So the average Hausdorff distance to the expert ground truth is less than 20 nanometers. One thing that we need to do to get that is to use this TAP method, tri-axis prediction. So we run the model over the data in all 3 directions, which fills in any missing gaps. And that gives us beautiful 3D reconstructions of the nucleus. And we're working on running the algorithm across lots of different data types from different microscopes and with different cells and tissues to make sure that it generalizes. And we've now done the same with mitochondria. So this is from our EtchAcell project Powerhouse Hunt, and using the algorithms from the nucleus to run on mouse and rat brain data, which was part of the Mito-EM challenge that was out recently. And what this all means is that we're starting to reconstruct whole organisms whole organelles at scale, not organisms yet. And that means that we can start to think, or I should just introduce you to the team, we can start to think about morphometrics. So we can start to think about converting our qualitative volume EM data into real quantitative results. So Martin was working for the deep learning with our scientific computing team at the Crick, run by Amy Strange, with Luke Nightingale and Joost de Volter, and also an undergraduate Sandwich student, Harry Songhurst from the University of Manchester, who joined us for a year. And I still find it incredible that an undergraduate could have done a lot of this work. So we had a second undergraduate student this year, Adam. He joined us from the University of Sussex and is one of the generation of master's students and undergraduate students who had to work at home for an entire year, but he did an amazing job. And with Martin, he worked on, so far I've shown you this part of the pipeline. So we acquire the raw images, we make the segmentations, and then we generate meshes. But what we're finding is that when you generate meshes in different software, like Fiji or iMod, you get a different mesh each time. So it might be fine for visualization, but it's not good for quantification. So to this pipeline, Adam has added a conditioning step, as well as a quality control step, which means that we can check the final condition meshes that can be quantified to make sure that we haven't changed anything from the original segmentations. So those are now ready for shape analysis, and that work should hopefully be bioarchived soon. And so we've closed a couple of the earlier projects, but we still have HSL projects online that you can take part in. We have HSL Virtual Reality up, which is a collaboration with Rod Parton and John McGee in Australia. And they've already worked together on animating volume EM data and creating these incredible cellular nanospaces that you can walk through and even sense different organelles in VR, which is being used as an educational tool, but I think is probably the future of how we interact with these data sets. And that was also done with Kerry Thompson and her master's student this year. And you can also have a go at HSL ER. So we're gradually moving through the organelles, and they're gradually getting harder for the citizen scientists to draw around. ER is an especially difficult one and requires people to sit down and to do more segmentation to get through the volume, because there's a lot of it in the cells. So this is working with Guy Pearson and Jez Carlton. I think I said that. And if you'd like to have a go, this is actually using their real data. So from a real project, looking at how organelles divide in mitotic cells. One of the things that came out of this for us is that you really need good reference or benchmark data sets that you can develop these image analysis techniques on. So for that purpose, we collected serial block face SCM datasets on HeLa cell pellets at 2 different resolutions, 5 and 10 nanometer pixel size. And those were used to feed out to citizen science and to do ground truth expert segmentation on. And we think it's really important to share this data so that others can use it as well, especially for the development of deep learning algorithms. And so you can find them on Empire, the Electron Microscopy Public Image Archive. And you just need to search for Empire 10094 or 10106. And you can find the details for that in Helen's paper with Martin and all of our collaborators on deep learning for automatic segmentation, the nuclear envelope. So the data is up on Empire, and you can also access all the expert and citizen science segmentations and the models through the GitHub links. And not only that, but we're talking about correlative light and electron microscopy here. And one of the really difficult things is lining up a 3-dimensional light microscopy stack acquired before sample preparation for EM with a volume EM stack acquired afterwards, because during sample preparation you get shrinkage and warping artifacts, and that makes it very difficult to have a good 3-dimensional overlay of the 2 data sets. And so Mary Charlotte in the lab and Chris Peddy have acquired 4-color AIRI scan data and then focused ion beam stack. And that's partially for our own development work. This is a project with a 3rd undergraduate sandwich student, Daniel Krenzel, and with Ricardo Enriquez and Roman Lane to try and automate overlay of fluorescence and EM data. But this data is about to go up onto Empire as well, with all the associated metadata, so that other people will be able to use it as a benchmark data set. And again, hopefully that paper will be bioarchived shortly. So I've just told you that it's really difficult when you have a 3D light microscopy stack collected before sample prep to line it up with your EM stack from after sample prep because of the shrinkage and warping that happens in the middle. And that affects the precision with which you can do correlative light in EM. And so I'm going to tell you a little bit about how we overcome that and what we're planning next. So Chris has developed this InResin fluorescence protocol, which is based on work from both Susan Nixon and Kent MacDonald and Rick Webb, with a few slight tweaks. So we take the samples and instead of fixing them with chemicals, we high pressure freeze them. And then while they're still at cold temperature, we substitute the glassy ice in the sample for a solvent like acetone, which contains a heavy metal. And in this case, we avoid osmium because it blocks fluorescence. And we use uranyl acetate instead. And we also maintain a small percentage of water because GFP, YFPM cherry need a hydration shell to fluoresce. And then we go into a methacrylate resin, which will tolerate a small proportion of water and has a very low autofluorescence itself. And when you do that, you preserve the fluorescent proteins in the block face. And when you zoom in, you can see cells. And you can still see here M-cherry H2B and GFP-C1. And that's labeling diacylglycerol and cell membranes. The big problem is that because you're only using a very light heavy metal staining, your samples no longer go black, which is something that us ultramicroptomists use to tell us where the cells or tissue are in the block face so that we can trim down to them. Actually, the only way you can see your cells in the block face is to put them under a fluorescence microscope. So cutting a very long story short, the microscopy prototyping team in the lab designed a miniaturized fluorescence microscope and integrated it into an ultramicrotome. And just having a quick look at how it works. So we can illuminate the block so you can see the cells to trim down to the region of interest. We can then trim using the Ultra LM, and I'm just going to move through to the point where we're cutting sections. So we bring the Ultra LM into position and align it to the block face so that we're focusing on the cells, which you should be able to see up here in the right-hand corner. So this is now focusing on the cells. And then once the run is started, the sample lines up with the microscope, then it passes down over the diamond knife, cuts a section, comes back up and over on its return, and then you can see the flash of fluorescence images taken. Again, you take a section, the sample comes up and over. Oops. There's a flash when the image is taken, and then it comes down. So one of the challenges here is fitting this kind of miniaturized light microscope into the ultramicrotome. The diamond knife here is worth about £2,500. So you really don't want to hit it with a big bulky objective. And you don't want to introduce any shaking into the system because otherwise you're going to affect your cutting. And then when you're ready, once you have a nice ribbon of sections, then you can move the system out of the way, come in with your grid, pick up the sections, and then put the Ultra LM miniaturized microscope back into place and continue monitoring for fluorescence. So there's a couple of really interesting things here. So the first is you're effectively doing serial block face fluorescence microscopy rather than serial block face scanning electron microscopy. And here you can see we're shaving tiny sections off the front of the block and gradually moving through the cells. And then something else that the team noticed, particularly Martin, was that when you focus at the block face and the cell, so this would be a cell that's close to the block face, but if the cell is much deeper, then when you image at the block face, you're effectively kind of imaging the point spread function at the block face. So you're imaging a circle. And you can use that to predict when the cell is going to reach the face of the block. So as you cut away sections, the circles get smaller and smaller until you reach the cell, and then it's cut away. So if you turn that kind of point spread function on the side, then you can see it's a funnel, and you can use a line of best fit to predict when the cell's going to reach the surface of the block. So this work means that we should be able to automate the process of picking up only sections that contain the fluorescent region of interest. And this was done by Martin with Lizzie Brahma, who's left the lab, and Wei Guan, who has come in to work on the project, and also with the support of Chris and Azumi, who've been testing the system and making sure it's user-friendly. So once we've picked up those sections that contain fluorescence onto grids, then you can image them in separate fluorescence microscope and then go straight into a TEM. And in fluorescence, you're imaging sections that are below 100 nanometers thick. So you're already working in super resolution in Z, and you've removed all of your out of plane fluorescence. So the overlay of the two signals is very good. So here you can see that diacyl glycerol is sitting on the nuclear envelope, on vesicles in the cytosol, and at the tips of the Golgi apparatus. You can also image those sections in an integrated light and SEM. So now sections are being put down onto a larger substrate made of Indian tin oxide coated glass. And you can see you've got the light microscope with the laser coming up underneath the stage with the objective, and then the electron column coming in from above. And you can align the two beams to each other very precisely, because when the electron beam hits the glass, you get a cathodoluminescence signal, which can be detected in the light microscope. So theoretically, you can align the two within 50 nanometers of each other. And looking at HeLa cells infected with vaccinia virus, which has a fluorescent YFB core. So each of these is a thin section through a cell, and then each of these dots is a virus. When you zoom in, you can see that you get a very tight overlay of the fluorescent signal to each of these virus particles. And then cutting another very long story short, something that's super cool is that when you look at GFP, RFP, M-Cherry, etc., in these InResin fluorescent sections, you turn the laser power up just a little bit, they blink like crazy. And you can do that in atmosphere, or you can do it at 200 pascals of vacuum with water vapor. And you can see the blinking compared to just whole cells imaged with the same laser power and an anti-fade mounted. So we can use single molecule localization microscopy reconstruction to improve our resolution. And again, looking at lower magnification, each of these is a cell with virus particles. This is the super resolution light microscopy reconstruction. Then we switch directly to EM and do the overlay. We can zoom in, so wide field, super resolution, EM, and overlay. And you can see now that the signal is shrunk right down to the core of the virus particles, which are about 250 nanometers in diameter. And with Susan Cox, we did some calculations, and we think we have a localization precision of around 80 nanometers. And in the future, we'll be aiming for around 50 nanometers cubed, 15 nanometers kind of voxel accuracy. Just to finish up, there's a couple of challenges here. The first is what we call the contrast compromise. So this is cutting sideways through the pellet of HeLa cells, and up here was against the metal during the freeze substitution process. And down here was up against the solvent with the heavy metal. So you can see this side has good electron dense staining on the membranes, and on this side it's very poor. And our best fluorescence preservation is away from the area of good membrane staining. So there are various potential solutions. The first is to overexpress the protein of interest, and actually some labs do this where it won't affect the biology, but in other cases it's not a good option. So here with diacyl glycerol at the top, we have poor membrane contrast and good fluorescence, but down here we have much better membrane contrast. So we're looking at a cell that's really overexpressing just to be able to see the fluorescence. And the ER membranes are wrapped up because we've screwed up the biological system. Another option is to find a heavy metal that is more compatible with fluorescence preservation. And Chris Peddy worked with Sangeetha Hari from Delmic and investigated a whole load of different heavy metals. And to cut a long story short, again, uranyl acetate is still the only good option. What is looking most promising is these new osmium-resistant fluorophores, like those developed by Gabby Paez-Segala and Lauren Luger at HHMI Genelia and now at UCSD, which survive some heavy metal staining. And there are actually more papers coming out on similar probes now. So this is looking really good. And then the last challenge that I'm going to go into here is taking the technique into 3D. So we're not limited to looking at the thin edges of cells. We can actually cut sections through the center of cells or through anywhere through tissue. So if we then start taking serial sections and we image our fluorescence and then our EM in each of those sections, then we can build up completely matching stacks of super resolution light microscopy and serial EM. And what we're planning to do next is then to extract the organelle maps and to overlay our 3D localizations of the proteins and to start to quantify differences in health and disease and in interacting organelles in volumes, but with high precision at the same time. So bringing together everything I've told you about so far. And again, the elephant in the room then is the data analysis. So these techniques are throwing out massive quantities of data, and the way that you process and handle these in 3D is not known yet. And that's actually what we've just been funded to do with the ChanZuckerberg Initiative, with a very exciting team with Paul French at Imperial College London for his open frame microscopes and his expertise in single molecule localization, with Ricardo for his expertise in single molecule and also in data processing, with Martin and Amy at the Crick, and then with Ben Lewes at the University of Stellenbosch, who is an expert in correlative microscopy, and who is, so one major aim of this is there's no point in us developing this if it's going to stay stuck in our lab. So these advanced imaging technologies are amazing, but if they're only available to a few researchers to answer a few or a handful of research questions, then it's not really reaching its full potential. So this pipeline for high resolution CLEM that we'll be developing should be, and that's a massive aim and a massive challenge, deployable in any light microscopy facility without the need for an expert electron microscopist. And that's what we're going to be working on for the next 2.5 years. And Ben is going to be really field testing that system for us and making sure it works outside of our well-equipped facility full of expert electron microscopists. And a key part of that then is going to be new training methods. And this is absolutely essential for rollout of advanced techniques. This effort is being led by Rafa Karzeniga, who's deputy head of the facility, with Catherine McLaughlin and Chris Peddy. And there'll be a video released online showing the way in which they're starting to develop new videos, new methods to train people here in volume electron microscopy, which will complement open source protocols we have by really showing people the key things that you can only see when you watch an expert do this and that you don't get when you read a protocol. So with that, I'm going to wrap up. So I basically mentioned everybody in the lab and most of our collaborators throughout the talk. I'd like to thank the scientists at the Crick who we collaborate with as well. The only person I didn't get to mention from the team during the talk is Aaron. So I've just popped his picture here. So a few of the lab have been helping with the COVID efforts in the last year. Alana's been on the testing pipeline, and Rafa and Catherine have been vaccinating people, and Aaron has actually been officially seconded over to the COVID pipeline for the last year or so, and we've just got him back. So next time I'll show you some of his data. And please keep an eye out for adverts for positions, because we're going to have CZI positions being advertised soon. And thank you to our funders, and finally to everybody who's contributed to EtchACell. So we have an extra 6,000 or so people in the lab now, and I'll just leave that running while we take questions. Thank you very much.
Lucy. Oops, here we go. Thanks very much for a great talk. I think when we get the sort of questions sorted out, I'm just going to run the quiz. So I'm just going to share screen to get the Mentimeter quiz. The link is in the chat. So just have a look at that. Let me get it up. There we go. All right. So, all right, okay, we've got quite a few participants already. That's good. A few more entering. I'm just going to wait a minute or two while everyone joins in. All right, so just to remind you, you want to go as quick as you possibly can to have a chance of winning, and you get to win the magnificent fold scope as our prize. Let's see where we are. 22 people in so far, 23. 24, they're still coming. All right. Okay. Aye.
Nick, you need to reset and start from the beginning. So. I think you're giving tough competition to Stephanie.
Come on. Yeah, here we go. Right. You can put your humdinger. There you go. That's what we've got so far. Someone's going for EM. Distinction is no longer relevant. I think probably where we're heading. That's my personal view. All right. Everyone, back here again. All right, let's go. Let's start. Still getting people joining. I'm just going to have to hang on a minute until a few more people join in, because I can see the counters going up.
Yeah, we were at about 20 something before.
All right, here we go. Let's run it. Okay, question number 1. What's the smallest slight thickness commonly used in focused ion beam scanning electron microscopy? Is it 1 nanometer, 5 nanometers, 50 nanometers, or 5 microns? So answer as quick as you can for most points. And 14 people have gone for 5 nanometers. Yeah, okay, that's good. Let's see what the scores are like. Oh, it's pretty level pegging actually. MC is winning. Let's see what happens next. But I think this is still open competition here. Next one. What does IRF stand for? Internal reflection fluorescence, international radiation facility, InResin fluorescence, or instantly recognizable faffing. That is definitely what goes on here. InResin fluorescence. Okay, we've still got 14 good contenders. And all right, let's see who's winning. Patricia has now jumped up to number 1. MC still hanging in there. Okay, good. Question 3. Sorts of which heavy metal are compatible with the preservation of fluorescence for high resolution CLEM? That's osmium, metallica, lead, or uranium. Which one of those is good for fluorescence preservation? We've got uranium coming top. Let's see. Oh, there's still neck and neck at the top right. This last question, you've really got to be quick now if you want to win. Why is it important to? All right. Well, I didn't even get a chance to read it out. So, all of the above. Anyway. As a resource for computational scientists to design and test. I'm not going to read it all out. We've got nine votes here who are very, very, very quick. I think what we're suffering from is internet lag. Yep, Tricia, you're the winner. If you could just make yourself known to the organisers, just stick your email in the chat or something like that, and then we can send you the prize. Okay, with that, I'll stop sharing and go back to all. So,
considering how popular Squid Games on Netflix is becoming, we should say that who didn't win is safe. Okay, nothing bad will happen to them.
Yeah. All right. Questions?
Yes, so we can go for questions. So yeah, so everyone, we allow you to ask your questions yourself. So I see like Alex Minrove have some questions. Alex, do you want to ask the question yourself? I can't see everyone.
Hi.
Hi, yeah, go ahead.
Just a thought, it's just nothing like a question. Very often you can do just serological estimates, especially the volume. And it's quite easy to make a point counting or intersection line counting. And you don't need to trace everything. It's like a bypass, like a walk around all the problem that you need to trace very complicated structures.
Yeah, I completely agree with you, Alex. And we do, stereology is really important, and we do still use stereology for some of our work. So what I haven't really talked about today are the projects, and there are plenty of them where we really just still use transmission EM, usually correlative, but still just 2D transmission EM, and we can still get a lot of information. Even in 3D, I think Yannick Schwab's work showing kind of stereology in 3D as well by imaging parts of cells with focused ion beam SEM is really important. But there is this tantalizing view that in the future, when we can segment things automatically and then maybe downscale them and then maybe turn them into numbers and eventually, can't imagine it, but throw away the raw data and just return numbers back to the researchers, then I think we're entering a different era when bioimaging and electron microscopy becomes much more useful for answering scientific questions. So it's all part of a journey, I think.
Thanks.
We still have one couple of minutes, so if no one is there, I can ask. So Lucy, what's the advantage of the super resolution data? So we see blobs, and as predicted, we see in where they belong, and they are still like about 200. So there's no internal structure to it that would add extra layers. So how do you see like super resolution actually helping EM data, or do you think it's just a proof, that something exists? So, yeah.
Yeah, I think structure on its own can reveal new insights, but it's much more powerful when you know which molecules are there and you maybe have a dynamic history of the sample from the correlative as well. So the point of the super resolution is that you really shrink down your localization. At the moment, we're lucky if we can localize to a compartment of maybe half a micron in diameter. So if we can say, we think this fluorescent molecule is in this endosome, that's quite hard already. But if we can get down to 50 nanometers, we're starting to approach the resolution we get with double antibody labeling with a gold particle. And actually, you might be able to start to say, well, there are a few of these proteins clustered on this tubule coming from this endosome, or this protein in this disease is clustered between the endoplasmic reticulum and mitochondria at this particular site, and we can disrupt it by treating with this drug, something like that. And to do that, you need higher resolution.
Yeah. We have one more question from Abigail. Abigail, do you want to go ahead and ask the question yourself?
Sure. Hi.
Hi, Abigail.
So great to see you, Lucy. I just love your talks. I always feel inspired and regenerated, I guess, after talking to you. So early in the morning here for me, so I'm a little disheveled.
What time is it?
Well, it started at 8. I'm not an early person, so. So, I mean, everything is just wonderful to see. I'm especially curious about the InResin fluorescence. I mean, as you say, the precision there, given everything that you just made a comment about how super resolution is still getting there, the ability to have precision is really where we have so many people coming to us and just sort of like just tugging on our elbows for a little bit more.
Yeah.
Where do you feel the InResin fluorescence is in terms of, is this something where we can start applying it in a routine way to people's samples, or does it still really require a lot of refinement to work out the tweaks?
I think it's very close. So I think the key to this is going to be getting just a bit more contrast onto the membranes whilst keeping most of the individual protein fluorophores active and emitting. And I think these new probes are going to do that. I think once we see that, then actually there's not so much point necessarily in doing the fluorescence first, or you do your fluorescence first and get the dynamics, and then you preserve the fluorescence every time so that you can increase the localization later on. But that contrast compromise has to be overcome. And at the moment, the other thing is there's a high pressure freezer in the process, which, as you know, can be finicky and require a lot of repetition to get the perfect sample. So, and then of course, ultramicroscopy, which is hard and limits these things to expert electron microscopists. So we're going to try and take all of that out when we develop the pipeline and make it as easy as possible. And it's a big challenge, but we think we have the ideas and the people on the team to do it. So check back in 2 years and fingers crossed.
Absolutely. Thank you.
I know we are very tight on time, but we take one last question from Catherine. It's on fixation. So Catherine, if you are there, please unmute and go ahead with your question. Yeah.
Yes, hi. Yeah, I'm very curious to know about whether you use alternative fixation, simply because with tissue samples, you often use formalin rather than paraglutaraldehyde. And I know that depending on the fixation, it can change what you see in your images sometimes. So your protocol used one, but I'm curious what your view on this is, since not all samples can be fixed with paraglutaraldehyde.
Yes, very good question. So if we're preserving the fluorescent proteins, then we leave out the glutaraldehyde. So it's paraformaldehyde only. And then after we've done fluorescence imaging, we add in glutaraldehyde, or just use a very low percentage. Formalin can be problematic, especially if you're talking about preserving biopsies, because some of the formalin makeups include methanol, and methanol is really bad for EM because it strips out all of the membranes and disrupts it. So whatever you're using, try and make sure that it's neutral buffered formalin that doesn't contain methanol, and then you should probably be okay. So there are different ways to do it, and every EM facility has a different way of fixing. General principles are, if you want to preserve fluorescence, avoid low traldehyde, coming up with it later. Just avoid the methanol.
Thank you so much.
Yeah. Thanks, Lucy, again for this excellent talk. I think this is one of the best talks we had in Imaging One World. So thanks again. Like
thank you.
it was very clear and yeah,
Thank you for the
very nice talk.
invitation.
Yeah.